Good evening, everybody. Today we'll touch very important point, DNA methylation, and you'll see also KABUKI syndrome. My name is Alexander Kell, and you're at the coffee breaks with TRANSFAC. Basically, take your coffee, relax, and enjoy the presentation today. All right, so let's go on. So as I said, the topic today is DNA methylation, but let's start from here. All right, so I want to show you this very nice central dogma, which was made by ChatGPT, and then now we have to understand that this central dogma is kind of changing because we know that epigenetics and generally epigenomics, if we talk about the whole genome changes, is a kind of agent which comes in the night to this big library and actually rewriting the books all the time. So, and this gets completely different. So you basically open the book today and it's something written in there, but then tomorrow it can be a different thing, which is written in the same book. So that's, what we have to consider when we talk about epigenomics. And, but what do we mean? So what do we mean rewriting the book of the genomics? Of course, the genetic code is not written. So if you have a gene which is encoded there in the genome, this gene is most cases is basically transcribed and translated into the protein, which epigenomics is not basically changing. What is changing is whether this gene will be in fact expressed or not. So the regions of DNA are big regions of DNA in the chromosome where all these regulatory messages are written, and those regions actually target for these epigenetic changes. So this little agent is actually doing a lot of changes in the chromatin by doing the histone modifications, which leads to different behavior of these histones, and they are able to open or close different regions of the genome. Then transcription factors can access those regions. And there is another topic which is important today is DNA methylation. So actually these two things are talking to each other, and we'll touch this today. But basically, I would say the DNA methylation is more solid changes of epigenomics when this DNA is methylated, and this methylation status can be even go from one cell to another through the cell division. Although the histone modifications, they also can go, can also transit to a certain extent through the cell division, but most of the time their changes in histone modification are more rapid, and DNA methylation is more solid. So basically, it's kind of if you can write in the book just by some pencil, or you write it with a pen, with a real pen, and you write it down. And the next level of changes in DNA are actually mutations, which are already genetic changes. So, and DNA methylation is still considered as epigenetic because it can be changed back. And if we look at this from chemical point of view, it's it happens in these dinucleotides, CG dinucleotides in DNA. It's always like that. And then the special enzymes, they just put this methyl group on the C in a particular position. And then this group, this nucleotide becomes a different chemical structure. And since it's a different chemical structure, it can behave differently in DNA, compacting, and also will behave differently in the interaction with binding proteins. And that's what we are going to talk today. And of course, there's there's also enzymes which remove this from the cytosine and then reverse this methylation back. And why DNA methylation is important? So I listed here some important topics. There's just actually 3 topics are important. First is, as I said, this DNA methylation can influence gene regulation. So why? Because basically it's changing the structure of DNA. Cytosine gets this little group, and it influences the binding all kind of protein machinery, which actually read the regulatory code. We will talk about this in detail later on, because that's a major topic for us. And because of that, because of these changes in gene regulation, it influencing cellular differentiation. So differentiation process, differentiation processes can go differently. And of course, this very practical thing, those methylation patterns of DNA can be used as biomarkers to detect diseases, to detect special cell types, to detect tissues, and so on. So these are 3 important topics. And if we talk about gene regulation, it's actually very simple. If you have these CpG places, sites in DNA, so simply DNA has always some nucleotides, and there are some pairs, CG, and there can be target for this methylation. And some of them are methylated, some of them are not. And actually, generally, in the eukaryotic, well, in mammalian genome, that most of the CG actually are methylated. And then, well, there is very rapid process of methylation and demethylation, and we'll talk about this a bit later. But then what you see, and it's a kind of general point of view and kind of a dogma right now, which we also will address and question, is that if you have a hypermethylated region, so basically those CpG sites which are methylated all the time, or methylated in most of the cells which we are studying, then they're hypermethylated, which means their methylation has a higher frequency probability. If you pick up a random cell, this position will be methylated, then in a random chance, right? And there are kind of hypomethylated. So those places, those sites of those dinucleotides, CG dinucleotides, which are methylated less than average, and simply methylated or not methylated. So basically in average methylation status. And it's a general belief that if it's methylated, it leads to the inhibition of gene expression, and hypermethylated. If it's a hypomethylated, it goes to the activation of DNA, of gene expression. And that's, this observation was made when people were looking at these points, which are called CpG islands. So basically, CG dinucleotides, they tend to be methylated because enzyme is there and it's active, and it's tried to methylate everything. But due to important process, which we'll touch a little bit later, evolutional process, since this methylated, cytosine can be converted to thymine, and then this can lead to a mutation. Then evolutionally, upon the evolutional pressure, the CG dinucleotides are actually diluted from the whole genome. There are less CG dinucleotides in mammalian genomes in numbers, in frequency, than you can observe by random chance if you just count number of C and G nucleotides. You can multiply frequency of C by G, and then you can calculate how many CG pairs you can expect. But if you count how many pairs you see on genome, you see they're much less than expected. And that's because of that evolutional pressure. So this CG is kind of bad because they can lead to mutations. But there are places in genome where those CG dinucleotides are kind of grouped together, and they are sort of islands of CG. In those regions, the number of CG dinucleotides is higher, actually, a little bit higher than random chance. Sometimes it's approaching the random chance frequency, but still it is higher than in average frequency in the whole genome. So that's kind of interesting because it goes against the overall evolutionary pressure. It means these CpG islands seem to be evolutionary advantages. So they're kind of a selected features of genome because many of them are located in the promoter areas, in enhancer areas of genes. And it was observation when they are hypermethylated. So many of them are methylated together. Many of those methylations, CpG dinucleotides are methylated altogether. Then these genes are actually repressed, which are located near those CpG islands, or well, the start of transcription actually is just on the CpG island. And that's kind of lead to the observation that in general, hypermethylation should lead to gene repression. Okay, so, but first of all, I mean, there is, again, what I said about these CpG islands. They're in general, CpG islands, CpGs are quite low frequency, but there is this definition of the islands. If you see that, the number of frequency of CG is higher than expected frequency of CGs by random chance, so when you multiply two frequencies of C and Gs, and higher than 0.6, so not very much higher, but a bit higher, and this region is extended longer than 200 nucleotides, this is defined as a CpG island. All right, so even very long ago. This definition was from 1993. And the reason of dilution of CGs from genome, as I said, that C can be transformed into T due these chemical reactions. And then if you have this, instead of C, you have a T in that position, it will mismatch during the cell division, and can lead to a mutation. That's why these generally CG dinucleotides are diluted. But actually, this general concept and dogma that actually CG hypermethylation and in general CpG islands are there to repress genes is now getting a little bit, revised, because it's not as simple as one can see. Because if you think about the gene regulation, gene regulation, you cannot avoid transcription factors. So you have to consider transcription factors all the time, because the gene regulation, the major machinery in eukaryotes, which was developed in evolution for gene regulation, is based on transcription factors. And if you have to repress or activate the genes, you have to play with transcription factors. You have to, attract activators to activate the genes, or you have to attract repressor to repress the genes. Of course, there are other mechanisms, and basically you have to really conserve the genes, conservate sort of, to repress it forever. For instance, the gene which has to be expressed in early stages of development, but for the whole life later on, it has to be silent. So then the other mechanisms are involved, not just recruiting repressors like transcription factors, which are kind of coming and going away, but involving these more solid mechanisms of compacting the nucleosomes together, and there the DNA methylation play a role of kind of conservation of that area. But in the fluctuating sort of situation, one gene has to be switched on or switched off in different stages of development, or during the cycle of cell cycle, or in many other conditions when the gene expression has to be regulated very dynamically. The DNA methylation play a role also of regulating these dynamic changes of expression. And all this regulation is based on how these transcription factors, which are involved in regulation of genes, are binding to their target genes, target sites. And these target sites may contain CG nucleotides, like this famous transcription factor E2F. Most important is E2F1. but here we see to have 8, which is also important. So these are transcription factors which are involved in regulating very, tightly cell cycle, and there you have to, play a lot of those binding sites. But those binding sites for these transcription factors like E to F, they contain these CG denucleotides, so which prone to be methylated, right? And if they are methylated, then the question, what happens with this binding? So whether this methylated C will abolish the binding of this factor or not, or maybe it will actually lead to even better binding of that factor. So, and this question is important, and the general belief, initial hypothesis was that in most cases, this this methylation of CG should actually abolish binding of the transcription factor. For instance, if you have a transcription factor E2F1, and we collected those binding sites from the literature or from ChIP-seq experiment, and build this positional weight matrix, which we have in TRANSFAC. As you know, we have now a big collection of those matrices in TRANSFAC. We have more than 10,000 matrices from many transcription factors and many organisms. And we cover, I recently calculated about 93% of all known transcription factors in human. So for each of them, or for each transcription factor, we have at least one PVM in TRANSFAC. So those PVMs, they actually, PVM is positional weight matrix. They are representing the binding motif for the transcription factor. And as you can see here, we have sometimes this CG repeat, even that happens in the site. So, and those matrices, as you know, are used to find new sites in the genome, in promoters, and they, with this matrix, you know what is the mechanism of binding of those factors to the sites. But what happens if this C is getting this methylated, so it may lead that actually factor is not anymore binding to this place. So which means we can say this cutoff, which we can calculate by the score of that site, which we can calculate now by changing this C in something different, in some less energy binding maybe. So then the score getting smaller, and then the site is lost. So that's a general concept. But actually, a very interesting paper and study was done recently, a few years ago, in Karolinska Institute by the group of UC Taipei, and that's really a fundamental paper, I must say, and somehow forgotten. I don't, but everyone should read this paper, because what they did, they asked a question: what happens if we mutilate the oligonucleotides and do this SELEX experiment? The SELEX stands for, for the random selection of oligonucleotides, which are in vitro, which are bound by transcription factor. So you have a transcription factor, and you put randomly, random oligonucleotides through the system, and then you check whether this factor is actually binding to a particular oligonucleotide, and if it's bound, these oligonucleotides are selected, and then they are actually mutated, and then there are several circles of those mutations and selections, and then at the end you get a pattern, so a number of those oligonucleotides which are selected by these transcription factors. So these factors tend to bind to those oligos, and then we can build positional weight matrices, the consensus of those among those oligonucleotides which are bound by this factor. But what he did is, in case of the if this motif is containing CG denucleotide, one or several, he would methylate them, and then do the same procedure, but with methylated oligonucleotides, and see whether this transcription factor will now select the same motif, or it will select a different motif. So it will, if it select the same motif, it means it actually bind to those oligonucleotides, even the C is methylated, right? But if it selects a different motif, it means its transcription factor is actually avoiding binding to the standard place as it would usually bind. So definitely then methylate C would probably limit this binding, and it will bind somewhere else. So then he tried to run it, and he found actually different factors behave differently. So for instance, if you take this HOX, no, let's talk first about OCT4. OCT4 is a very famous transcription factor. It's one of the most important factor in development, actually. And that's why maybe it has this very strange behavior concerning the CG nucleotides. It's one of the pioneering factor in all these, IPS cells and all these kind of cells, which when you change the, when you dedifferentiate cells back. So OCT4 is really important. But now when he did this experiment with OCT4, he found that actually OCT4 is binding to, it's preferably binding to the CG, which is actually methylated. No, wait a moment. No, OCT4 is actually binding preferably to the right. So the primary motif is containing no CG. So if you have CG, then it is not going to bind it. So it's avoiding binding to CG if it's methylated. But for instance, HOXB13, which is another, it's quite also important factor in development, it behaves other way around. It's actually prefer to bind to CG nucleotides when they are methylated. When they are methylated, it actually bind. So it's completely different from what we thought, that methylation actually helps this factor to bind to CG. And when it's not methylated, it's avoiding the CG binding. And when he compared, ran it for many factors, there was about, I think, 900 different factors, transcription factors. He found that there are a number of factors which are in the middle here, which have, has nothing to do with methylation. Well, most probably, these are only those factors which contain CG nucleotide in the center. So for many of them, it doesn't matter if it's methylated or not. But some for quite many factors, it's very important to have this CG being methylated, and then they actually binding to it with better efficiency. And then there is a number of factors which are which avoid to be bound by methylated C. And the mechanism of, for instance, for HOXB13, they even considered it very carefully and built this model. Then they found out this is DNA and this is HOXB13. And you see this C is methylated, and in the reverse chain, it's again methylated. So two methylated C's here. And this C is actually, methylated C is contributing to the energy of binding of that factor. So it's like to those nucleotides to be there, methylated. So which means that methylation is not always avoiding binding of a factor, but it can be attracting a binding of a transcription factor. And this concept of that methylation is always leads to the repression is basically wrong. So it's not, it's maybe it's definitely in the case of, hypermethylated big CpG islands, they, in most of the cases, they lead to the repression of the gene. But in the interplay of genes in a normal situation, they can either lead to activation or to repression, depending where they are, whether the methylated position are actually located in a particular binding sites for an activator, or it's located in a binding site or for a repressor. All right, so that's actually why it's very important to analyze individual CpG sites. Because as I know, many prefer to first to agglomerate them in CpG regions and only give importance to those regions when they see a lot of methylated CpGs one after another. there is a hypothesis that if they are so organized in such a cluster, they actually lead to real expression change. But in fact, you have to consider each position separately. All right, so that's another nice picture which ChatGPT give me. When I ask whether DNA methylation is significant in different human diseases, you hardly can read anything here, but the only thing you see there, there's a lot of pictures, which means a lot of diseases are touched by this CG methylation, and you can name them, and basically I listed here a few disease areas, I would say, where this methylation is really important and it's known. For instance, in cancer, it's known that hypermethylation of tumor suppressor genes is leading to cancer. Or maybe it's a secondary effect, but basically it's really correlated with cancer growth when such tumor suppressors like p53 and p16 and other retinoblastoma, they are methylated and they are not expressed, and then they are not suppressing the growth of the factor. In neurological disorders, it's well known genes which are methylated in different neurological diseases. Cardiovascular diseases also a very important area for methylation, and important. For instance, TNF alpha is involved in this methylation in link to such diseases as cardiovascular and others, and related to inflammation. And actually, we'll see today on the example of KABUKI syndrome, a story about TNF alpha. And again, so autoimmune diseases, actually, again, related to immune system, also, related to methylation. And at this point, I would like you to get some bit feedback from you, and we prepared a little questionnaire. Yes, you can see it now on your screen. So please take your time and tick here which disease areas you're actually working or you are interested in respect to the DNA methylation, or in general, if you are working in this area, and then just if you're working in several areas, tick several. It's just interesting also to see, whether what I'm talking about, different diseases, is something interesting for you, because maybe you're doing something completely different. So please take your time, and when you're ready, we'll see in maybe in 5 minutes. I will ask our team to show the statistics. All right. Okay, but we are going further. So we close this, and then there's a important, list of how to detect this DNA methylation and where to do it. What kind of material we have to use, and this we will touch. We have also a couple of questions related to that, and just two big things which you have to consider. So of course, the final answer to reveal the methylated positions in the genome is a so-called bisulfate sequencing, when you would take the whole genome, and do this bisulfate conversion. There is a chemical modification of DNA, so you sequence the whole genome once without it, and then you do it with this. And then by comparing sequences of two, you have to sequence your genome twice, actually. And then you see which positions are methylated with which frequency. You simply count reads, as usual when you sequence. And then this number of reads will tell you how often this position is methylated. And of course, that's very good and very efficient. But of course, very expensive, because you have to sequence your whole genome twice. To reduce the price, there is this famous, reduced representation bisulfate sequencing, RRBS, where you sequence actually CPG islands. And that's, of course, good because indeed the CPG island is, they derive majority of the genes, but maybe 50%, I would say, or maybe more than 50, but not definitely not all genes are regulated through CPG islands. So of course you reduce your cost, but you will not probably find the answer to your questions. And the alternative is which is much more efficient from a cost point of view, is to do these DNA methylation studies using methylation arrays. And you'll see this methylation epic array. It contains almost 1 million positions from genome, and then of course it's even more reduced than considering only CPG islands. But in fact, in some to a certain extent, it's maybe better because it's not only containing CPG island positions, but other regions as well. Although it's of course much less. It's you have 3 billion positions in genome, but here just 1 million. 3 billion. Well, how many CGs is much less, but okay, still it's less than the whole genome. Of course, you can do other kind of things like targeted methylating sequences or special other techniques. But the two major approaches are either you do this bisulfate sequencing or use the methylation arrays. All right, so these are these methylation arrays. So infinum methylation epic array, which contains almost 1 million position, and that's a very important tool to study methylation. All right, so how do we actually will analyze those DNA's regions in our study? So we would like to understand how this methylation is changing, actually influencing the regulation of genes, which is in turn is done through the transcription factor binding to their target sites. And of course, if you consider those methylated regions, which you found maybe with DNA bisulfate sequencing or just by microarray, so you get those regions which are hypermethylated, which means they are the methylation, this methyl group on the Cs in those regions, in those places, in those sites are more frequent than by random chance. And then you can take those places, you may found them in your study, and maybe you found, I don't know, 1,000 of those positions or 10,000 of those positions which are hypermethylated in your conditions, in your disease you study. And then you can put those positions one after another and ask a question whether those positions are overlapping with a binding site for a particular transcription factor, like we were looking just few slides before. So when it's actually this CG is a part of the binding pattern itself. And of course, although of course this is just two nucleotides CG, right? So there are other, but binding sites is longer than these dinucleotides. So it means it contains other nucleotides left and right from this CG. So in some areas around this CG may be favoring to a particular binding site or not. So, and then you can compare. You can say, this is my hypermethylated sites, and here are the simple CGs, which is randomly just taken from genome, and they are kind of randomly methylated, average methylated, not hypermethylated, not hypomethylated. So this would be our control. So, and then we can count simply how many cases we can find a particular site overlapping with these nucleotides, and how many cases we find this site in the no sequences, in the control sequences. In fact, what we will do, we will extend this area left and right even more than the length of the nucleotide, of the binding. Why? Because, and that's an important point, these transcription factors, they are not binding alone. For instance, this factor is binding here, but it can interact with another maybe factor which is binding to the methylated CG. And through the factor interaction, they can compensate this binding. So that's why we have to look around to a certain region around the CG positions and calculate the number of sites found for one particular factor in that area and compare it in the regions which is around non-methylated sites. And there's a, you can simply, what we are doing, we count those sites. We then calculate the probability of, if we found, for instance, here, 1, 2, 3, 4, 5 sites, but here only one, then you can estimate probability that this 5 is higher than 1, whether it can happen by random chance or it's very rare case. So we can estimate the p-value of such a case. And then we do it for all 10,000 basically motifs from TRANSFAC database. And then we can select those motifs which are enriched in the areas around CG methylated, hypermethylated positions. All right, so I have in Q&A a couple of all right, so somebody is checking whether a GC 5 to 3 prime end or CG is 5 prime to 3 end. Yeah, right. So that's important to keep in mind. Maybe I made such a mistake. All right, let me see. But that's important. Well, we see it here. Of course, this is CG. All right, so like on this slide. All right, so we have here. You're right. So the CG. All right. So I think these guys, you have to either, in fact, to have it in a regular view, you have to inverse this picture, because indeed this it must be CG from 5 prime to 3 prime. And if you have GC from 5 prime to 3 prime, this position will not be methylated. Only position which is which is CG from 5 prime to 3 prime. So it's like in this picture. So you see it's from 5 to 3 CG, right? Okay, so all right, so we are still here. So we want to estimate the frequency of the binding sites and which are around the CG positions, and estimate their enrichment. Okay, but if we even go on further, and you probably have seen this in our previous talks, that important is actually cooperation of factors, and I will not stop here for a long time. I just tell you that we have a algorithm which checks not just each individual motif, whether this individual motif is enriched in the regions of interest, but it looks for clusters of motifs. Those motifs which are clustered together, so they are co-localized in a particular window. So this window may be around like 100 nucleotides, maybe less, so kind of a narrow window where transcription factors, when they are binding to these neighbor sites, they can interact with each other, and this compensate their binding, and they can bind synergistically. So we have such a tool which can find those compositions of sites. All right, so what I have a couple of examples where we apply our tools which calculate the frequency of binding sites for transcription factors in these CG regions, and then we get a very interesting discovery of the mechanism of the disease, or in one case, or we found actually biomarker for cancer. So if you have time, we can actually talk about those examples. I put them here in the slide. So one example is about colorectal cancer, and another example is actually about very varicose vein disease, which is very important. And but today I want to show one example online, which is about KABUKI syndrome. And KABUKI syndrome, let me show this a bit, is very interesting. Well, why we consider this KABUKI syndrome? Well, because we have a project running, funded by you, Marie Curie project, on KABUKI syndrome. It's very important because it's a rare disease, and I would say not much money is spent by companies in such kind of diseases, because you have in the world maybe only not more than 30,000 people which have this disease. But from a point of understanding what is a mechanism and maybe even helping those people, it's very important. That's why it's funded by such organization like EU. And the KABUKI syndrome is actually a mutation of one gene, in fact, two genes. It's a genetic mutation, hereditary, and then those people, actually children, they are diagnosed initially because they have certain face features which are similar to the Kabuki artists from the Kabuki theater in Japan. So, but actually, this syndrome is happens because of two proteins, which are components of so-called compass complex. It's important complex which actually were in fact doing epigenomics, but very sudden, very important and not very hard things. It's in fact doing a methylation of histones. And two types of methylation of K4 and methylation of H3 histone, and in position K4 and in position K27, two different proteins. And if they got mutation, hereditary mutation, then this function working not very well. So this methylation of these histones is getting not so well, going not so well, or kind of distorted. And since it's influencing a lot of genes, in fact, what happens, this methylation of histones is a kind of marker of activation of genes. So it's a marker of a promoter, active promoter. It's a marker of active enhancer. And those genes suddenly, get less methylated than needed or more methylated than needed. So this methylation function is distorted, and it's affecting the whole genome, actually, a lot of genes. Still, those people, they can live, and although they have many different symptoms in the various types of parts of body, in brain, in muscles, in heart, as I said, simply in muscles. Also, many symptoms are related to immune diseases, and that's what we will see also today. So the immune system is not working well. Basically, most of the symptoms are related to the development. So these changes of the methylation of histones is leading to the development defects. But it happens that this histone methylation, which in fact is regulating gene activity, is very tightly coupled with DNA methylation, although it's done by different enzymes. But when this is distorted, then DNA methylation is also distorted. Whether it's a direct effect or maybe a secondary effect, we don't know. Whether maybe this DNA methylation is kind of a response of the gene, towards this less efficiency of the histone methylation. This we don't know, but we see the clear correlation. And then basically, we took a paper from this, we took data from this paper where these authors, they took actually KABUKI syndrome and a similar syndrome, which is called Charge syndrome, which is have a quite similar symptoms, although it's a mutation of a different, another protein, which also involved in epigenomics. But today we just focus on KABUKI syndrome, and what kind of data they have. It's actually data done with methylation microarray. So where they did took a lot of patients, and then they took from blood cells from blood, and they study methylation in those patients. And then they have patients and they control group. So they have control 125 samples, and patients, they were like for the KABUKI syndrome, where this gene is mutated. This gene is mutated. We have just 11 samples. So we want to analyze that. And that's the point where I want to now to quickly go to our tools to show how to analyze such kind of data. We have a couple of questions. Yes, all right. So I think we'll touch this a bit later about OCT4. Did you come across with similar phenomena? Yeah, like, all right. So I think we'll touch that. All right. So, okay. So just wait. I will show how we analyze this data, and then we'll come back to your questions. All right, so what we'll do now, we'll look at this data in our tool. So this is our tool. This is a tool called geneXplain platform. And what I used a pipeline in this platform, which is called Genome Enhancer. And we are going to switch now to this pipeline. It's called Genome Enhancer. You will see this interface. And Genome Enhancer is a tool which allows you to put multiple omics data and analyze it. And actually what it's doing, it's not just analyzing DNA methylation. It's not just analyzing any kind of omics data to do statistics or things like that. No, we try from this omics data to find a cause of a disease. We want to try to find a mechanism. And in that mechanism, we even try to find we call them master regulators, and even go further to try to find maybe drugs which can influence those master regulators. So it's quite a complex pipeline. But what we will do today, we just, I showed it before, all the details, how we run it. But let me go to this project, KABUKI and charge syndrome. As you can see here, you drop your data. For instance, this list of hypermethylated CG dinucleotides. It's simple table, so you will see it in a moment. So actually, I have uploaded it. You see these tables they looks like this. For instance, KABUKI. So it has this 485 lines. So actually, half of those positions in the Illumina array, they gave a signal. And then for each of these CG dinucleotide ID, you have a value, which is a so-called beta value, which tells you whether the percentage sort of, it's a bit more complex, or if, value. Okay, you can say percentage of those CG dinucleotides which are methylated in a particular sample. So columns are the samples, the lines are these positions. And then you see those samples of from a KABUKI patients, and we have like 11 samples, and we have control samples here, which is 100 something, right? So quite much more. And then what we can do is simply when we put these two samples together into the Genome Enhancer. So let me go further. Actually, we form this experiment. So what we see, we have KABUKI samples, we can control samples, and then we go further. We define a disease, and actually, what kind of disease? So KABUKI syndrome is a very small syndrome, but it's just one line. But here, if we want to find a mechanism which leads to different symptoms, then we have to select different types of symptoms, whether it's immune-related symptoms or whether it's neurological symptoms. Depending on those selection which we do here, the analysis will go a little bit different. So it will put a particular focus on those master regulators which we'll found, which are related to that particular symptom or disease. All right. So, but what we will do now, we'll look now to the result. So here we have this KABUKI versus control. And we will open this result of the analysis by this Genome Enhancer, which, as you can see, what we run is we selected two symptoms, namely cognitive, congenital abnormalities and neurodevelopmental disorders. So very wide area of different symptoms which are important and they important for these patients. And then what the program is doing first, it takes those methylation positions and map them on different genes on the whole genome, and represent here this kind of picture where you see here the gene, the exon intron structure of the gene, and red bars show where you see this methylation, hypermethylation position. So it selects the first hypermethylated position, like 10,000 of them, and then it maps them to the genes. And you see many of Hox genes are here affected by those hypermethylation position. So the Hox, as you remember, we were talking today about them. These are transcription factors which are involved in development. So, and it's important, the KABUKI syndrome indeed related to the abnormalities in different types of development. Different tissues are developed a bit wrongly. So, and you see the different Hox transcription factors are involved in various lineages of development. Then it takes those genes which are affected by mutations and then mapping them to gene ontology, and you see that they're involved in cell differentiation, in cell junction organization, nerve development. So they are related to development of nerve, for instance, as we see. And what is important for us today is to see what was initial topic, how this methylation is affecting the binding of transcription factors. And what this program is doing, it's try to find such a combination of motifs which is happen most frequently around those hypermethylated sites. So how do we see this? Well, we can see it even visually. So if we see here regions which are hypermethylated, so this is a region which is in the center, has a hypermethylated position. And then this is a region around 1,000 nucleotide. It's like 500 or 600 left and 600 right. And then in this region, it found, you see, these little arrows. They represent, each arrow represent each individual binding site for transcription factor. And as you can see, those factors, they tend to cluster, those sites tend to cluster together in some clusters. And that happens sometimes in the middle of that area, actually near that position of hypermethylation. But sometimes they also located in a bit left and a bit right. So because those regions are still in the area of interaction of those transcription factors. So this tool can find those site combinations, which are indeed happens very frequently in those regions around methylation sites. And you see these histograms, they show how frequent those sites in the area which are around the hypermethylated sites compared to the areas which are around not methylated sites. And overlap is quite big, but still the significant discrimination and AUC is quite significant here. And then you see which transcription factor are actually important here. And I was really happy to see FOX and glucocorticoid receptor and other transcription factors, but these two are famous for regulating development. But then let me also go a bit further now, since from this analysis we know which transcription factors are involved in the regulation around those genes, around those methylation sites. Then what it will do further, it's built such a diagram which try to explain how these genes, which have this methylation region in their regulatory regions, in the promoters or in introns of those genes, how they are regulated by these transcription factors, and how these transcription factors in turn are regulated by signal transduction. And it identifies these so-called master regulators, which are shown here in red, which can be involved in regulating activity of all these transcription factors together, and respectively, many of those genes which we found hypermethylated. And as you can see at the top, we see here TNF-alpha, and that was interesting. In fact, not many studies I actually searched where it's known that TNF-alpha is related to the KABUKI syndrome, but that was the only factor, or not only, but it's one of the top. And when we intersect KABUKI syndrome, and that's another syndrome with similar symptoms, we again got TNF-alpha. And it seems it's one of the central mechanisms which is involved in many developments of the cells in the KABUKI syndrome. And that's maybe some new discovery here. Well, one important part here is that we even go further and look which of those master regulators which we found can be affected by a selected set of small compounds or drugs, which is quite practical for in some research to see whether, of course, you cannot completely cure this disease because it's it's monogenic disease and hereditary, but in many cases symptoms can be corrected by certain influence by some drugs. And for instance, one very big part of those symptoms are related to development of immune system, and that's definitely related to the TNF-alpha as a master regulator. It seems many immune cells are their development. TNF-alpha is involved in this development process, and such kind of symptoms related to the immune system can be corrected actually by some drugs. So that means these studies can also help to find a good cure. So that's kind of in brief how we analyze this type of data and how you see this kind of hypermethylation can be analyzed. Interestingly, that in this standard pipeline, we focus on hypermethylated areas. And that's also then today, actually, when I was preparing this talk, I thought, why is that? Why we, again, in that kind of paradigm that only hypermethylation is important, but why not also looking at the hypomethylation? So those regions, those sites, which are methylated less than in average. So which means they also affect some binding for other transcription factors, and they may show some other type of disease mechanisms. And I actually run this also for the hypermethylated sites. And here I have it in another project. Let me show you this one. And one important thing here is that, of course, I can just analyze the same, just hypermethylated sites as I did here, hypomethylated sites. So I just put them here and then analyze. I get another mechanism which may be related to different symptoms. So you see, we got here different genes and a different mechanism. So other master regulators are found, and other drugs are found. So you see there is a central of the mechanism, and other drugs are related to autoimmune diseases are found. So this is a more related, perhaps the part of the mechanism which is more related to the development of immune system. To find actually a more exact answers, it's better to combine different types of data And that's what I was trying to do here, but perhaps for that, we don't have much time today. So I was trying to combine expression data and the methylation data. And how to do that, I guess we'll talk about that later. Also, there was one question about that, but actually, just to open a little bit how to do this. So actually, when we analyze just methylation data, we map them methylated site on genome and we map them on genes. But in fact, we don't know, as I said in the beginning, whether this methylation is repressing the gene or activating the gene. We don't know. We just see that it's hypermethylated. And then depending whether it's repressing or activating, we should do different things. If it's activating and we want to, and this is master regulating, and when we want to cure the disease, we have to repress it. If it's repressing the gene, we have to activate. So it's completely different approach. So that's why it's much better to combine these two things with this methylation study with expression study. But for the expression study, you have to be very careful if you're selecting your material, because depending which tissue you select, you will find different expression. And for instance, here we choose data from which was where the authors were measuring expression on PBMCs, on blood cells. And then it's related to actually immune disease development, immune system development. And by combining these two type of data, now we can clearly see which genes are affected by methylation and they are upregulated, and which genes are downregulated, and then you clearly discriminate those mechanisms. All right, so I think with this story, I have to close because we actually have a couple of very interesting questions already in my list. Also, you are free to ask questions here. We have a bit of time. If you have time, I have time. So we can sit here and answer your questions. So don't hesitate to put your questions in Q&A. And in the meantime, I will look at my list of questions. Let me see. Okay, the first question is is the methylation pattern good to be studied by blood samples? Yes, that's a very interesting and important question. Actually, that's most of the studies. It is a topic. It's a material which people are using to study various types of diseases, because it's actually most easy to approach. So you get the blood samples, and that's it. So you have it. And then, of course, the question whether these methylation changes in DNA, they are reflecting those changes which are happening in the target tissue. Of course, the biggest signal which you get from blood samples will be those DNA methylations which are happen in blood samples. So, but there are types of, for instance, if you are interested in cancer, then you would take the blood samples and take from that sample those liquid so-called circulated DNA. So not the DNA from the cells of the blood, but DNA which is circulated not in the cells. So, and the idea is that the blood is actually so universal organ, sort of. It goes through many organs in the body, and also it touches a lot. If there is a cancer, if there are a tumor in the body, you get this blood around these tumors. And then if these tumors actually, if the cells of tumor are destroying, that part of the DNA is then leaking to the blood. And then you can detect it when you analyze the blood samples without actually cells of the blood. And in some sense, analyzing this material is kind of a mirror of all organs of the body, because the blood is everywhere. But of course, the concentration of those DNA will be not very high, and then you have to make a lot of efforts to enhance the signal. But that's possible. And for instance, in one of our studies, when we studied colorectal cancer, we actually did our study and analysis on the tumor samples, on the solid tumor samples after the surgery, where in the sample, we studied gene expression and the DNA methylation. But for the validation, we took actually a liquid biopsy, and we found that those markers which we found in solid tumors can be found in the liquid, in the blood samples, and they become actually quite efficient as biomarkers of early prognosis of colorectal cancer. So we have published such a paper where we actually list several of those biomarkers which you can find in liquid biopsy as a potential prognostic markers for the early stage of colorectal cancer. So the blood is important material. How to interpret DNA methylation changes at the single nucleotide level? Does it have a significant impact on the genes and diseases when it's super rare and in a diseased patient? Right. So exactly what that was topic today, and that was my claim, that it's very important to analyze it on a single nucleotide level, individual methylation sites, because as I said, even one site can change a binding for very important transcription factors, which has to be bound there, or is instead even bind even better if it's methylated. So it's important to analyze it on a single nucleotide level. But the question here is, in case if it's super rare, which means, as I understand your question, in your disease, or in disease which you study. By the way, we didn't look at the histogram of all diseases. Maybe my assistant can show it. All right. So here you see, it's good. At the end of the talk, I finally remember that. So what do we see here? Cancer and neoplastic disorders. So 48%. So the biggest interest from you guys. Neurological diseases, 19%, autoimmune diseases, 23%, metabolic cardiovascular diseases, infectious diseases. So approximately the same percentage of also aging is very important, and other diseases. So, but definitely the cancer and neoplastic disorders the biggest interest of you. Thank you very much. It's very important statistics for us, and also probably for you to see what is your interest. All right. So, in case if it's rare, okay. In case if it's rare, then of course that's is getting more difficult to analyze. So, which means you have to collect more of these positions, because what happens very often, you put your cutoffs very high, and at the end you get maybe 10, 20 positions which are super significant. And when you start analyzing it, it's not enough power to understand the disease mechanism. So what I would recommend, and what we are actually doing, is really lower your cutoffs of initial cutoff of selecting your individual markers, individual positions, because at the end, individual position is not really contributing a lot, but the significance will come when you combine those many positions together. So for instance, just by statistics, you will see that you select whatever 10,000, 100,000 positions, which have a little bit of difference between disease and norm. And then, a big percentage of them touches a particular binding site for transcription factor. Not all, but a significant enrichment. And this enrichment is statistically significant. So that's why you're still on a significance level, but you are not really cutting yourself from the very beginning by very high threshold, by initial significance threshold. So that's my recommendation. All right. So another question is online tools for the in silico DNA methylation, right? So that's my recommendation is our platform, geneXplain platform, and basically Genome Enhancer. And this tool is you can go to our website, and you can even register for it for free. It's a free registration. Of course, with this registration, you're not getting TRANSFAC. For TRANSFAC, you need a license, but the tool itself is ready. So all the tools which are there for your purpose are ready. So you can start analysis. You can use another library of motifs which you like, and do this enrichment analysis, do this composite model analysis, or even pathway analysis using some free pathway tools. But of course, to do it really in a depth, you can license also TRANSFAC and TransPath of signal transduction database, and use this tool online. So they are fully online. You don't need to install anything on your computer. All right. So another question: is DNA methylation mapping of each individual unlike unique like fingerprints, or are there some common methylation regions across the genome for each individual? Across the genome of each individual, mapping of each individual like fingerprints. All right, so I think I didn't understand fully the question, but I guess the question is about commonalities of this DNA methylation pattern between different cases, between different patients, between different individuals, even if they have the same disease. And whether, for instance, KABUKI syndrome, right, where the individuals have a different pattern. Indeed, they have, but they have an important commonality. And this statistical analysis, which is provided in our tools, actually help to find out these common parts. And if they would be completely random, you would not find this common part, right? So the statistic will tell no similarity. But since statistics found similarity, still there is a big variation between individuals, but there is a common pattern which you clearly see for a particular disease, for a particular tissue, for particular symptoms. All right, so these are questions which we get by email, but I guess I would also answer those questions which we have in chat. All right, we got them in chat, right? Not in Q&A, but in chat. Right, let me see. Right, so first question. Hi, interesting to observe that methylation might favor the transcription factor HOX, repress OCT4. Do we know that are they any of the methyl group edit like in histones have methylation 1, methylation 2, methylation 3, like many methylation. Thanks. No, of course, well, they're no and yes. So of course, one C can get only one methylation, right? So although there are different position of this loop where you can get methyl group, but normally it's get only in one position. So it's only one methylation group you get for one C. But what can happen, this region can have several CG dinucleotides in a stretch. So, and that like cluster of them. And then indeed you can have all of them methylated, or only one or two or three. And sometimes when the CGs are close together, they can even overlap for one binding for one factor. So some factors have their pattern where CGs are quite often. For instance, SP1 or even E2F. There are several CGs inside this pattern. And of course, this then become very important, whether it's just one C is methylated or all of them, all three are methylated, and then will influence, of course, the binding. I guess this is not studied well. So those experiments done by UC Taipei, he didn't actually check the number of methylation in case of there are several CGs in one stretch. All right, a very good question, actually. Another question, did you come across with similar phenomenon other than CPG, like 6MA, for example? Thanks. Okay, so okay. Right, so that goes even in more depth that even adenine can be methylated. No, well, there are not much studies about that, and actually, to say the truth, I don't pay attention too much about those very strange kind of methylations. but maybe they are important. So maybe it's a good idea to have a look at them. All right. So now we have all questions from in the chat so far. So if you have more questions, don't hesitate, put them in Q&A or even in chat. All right. So, and there is one big question here, which we got by email. A vast number of lifetime events and extraneous factors in every individual lifespan impact methylation in human genome. That's right. Many may have lasting effects on overall health, and some may be the underpinning for major diseases. When studying epigenomics in the brain of patients who died of suicide or other who had schizophrenia or bipolar disorder, how do we count for myriad of undocumented variations that may have subtle cause role in the overall disease outcome? Some of the true causal perturbations are never traceable under the heavy burden of medical effects, toxicology, and other comorbidities, and so forth. I would like to know your take on this. Yes, right. So that's actually not only a problem of this kind of very complex diseases, which you mentioned, like schizophrenia or bipolar diseases, which are very complex. That's true. And many things influencing this disease. But if you study cancer, if you study cardiovascular disease, you actually face the same problem. On lifespan, actually, individual get a lot of influence. They're eating different things. They're, they actually lifestyle is different, and all this is influencing the epigenomics of those people and changing it. Still, statistics can help. And it's really wonderful how statistics can reveal the little things which are out of the noise. So for instance, you have a huge noise, but as you see in our pictures, we have two histograms heavily overlapping, which could say, okay, you don't really discriminate them well, but they are significant. And which means, since for us it's not important to discriminate them completely, but really to understand the major driving forces. And then we can, of course, try to treat it. But as you are right, so by treating this will affect only small subpopulation among those individuals mainly, because only for them this driving force, which we discovered by the statistics, will be the major one. And the answer usually is by splitting. So then you take all your population cohort and try to cluster them by different means. Of course, you always cluster them by symptoms, but maybe even better clustering is by molecular clustering, when you would get subgroups, and each subgroup, although symptomatically they are the same disease, but this disease is caused by different molecular mechanisms. But doing the correct molecular mechanism clustering will help you really to find driving force in each of that cluster, and then your treatment will be then more specific. Finally, in the final goal is to do the personalized medicine when each individual is kind of a single cluster, which may be very far going dream, but at the moment it's the major approach is clustering patients into subgroups and then treating them each subgroup by special means, and that's going well. I mean, even in the such complex diseases like neurodegenerative diseases or neurological diseases. For instance, we have project on Parkinson disease, and we had also studies on Alzheimer's disease, where we try to actually separate and cluster patients into subgroups. We haven't done with schizophrenia, would be very interesting. And actually related to that is question number 7. which changes occur on the methylation on some genes during the meditation, or and that's was very good question for me. Actually, it triggers me to look for the data. And I found a number of studies where people were doing meditation, and they were studies to check the methylation status of those people before they do meditation or and after. And I analyzed this data, and I got very interesting results. I think I will make a separate coffee break for this type of data. It's interesting, right? So even such a very subtle things like meditation can influence your DNA methylation. All right, so I have another question in Q&A that was answered. Is it possible for G quadruplexes to be methylated? If so, what would be the result? G quadruplexes, what do you mean? G quadruplexes. Actually, probably I'm not aware of something very important. I'm sorry for that. I will check that, or if you can maybe specify a bit more what are these G quadruplexes. I didn't know, sorry. Okay. So another question in the chat. There, thanks. And I have here was the last question. Oh, no, there are also a few questions here. How can we solve the gene-specific hypermethylation to treat cancer? All right, so that's exactly one of the major driving force of the current studies and also our efforts to approach the cancer treatment on the basis of of epigenomics. As you know, the cancer now claimed to be the epigenetic disease. Although there are big discussions, what is the first, epigenetics or mutations? And there is a big, influenced people telling that actually epigenetic first. So first the sample, the cells are transformed, epigenetically to a state where they start proliferation, uncontrolled proliferations, and only later they got this is fixed by particular mutations. In a grown tumor, in a grown solid tumor, you get a lot of somatic mutations, like plenty of them, and because during the growth, this tumor evolutionally find a way to even grow faster. And for that, there is a quick evolutionary process going on in the tumor on the basis of genetics. But the very first step is now believed to be done to happen actually on epigenetic level. And if that's true, then it can be also treated epigenetically. So these cells, especially those cells which are going, I mean, this mesenchymal transition, which is the first step of cell transformation towards tumor. This can be maybe reversed by special treatment. And that's why when you have methylation, DNA methylation data on early stage of cancer, and you can apply such tool like I presented today, and then you may find the master regulators which drive this process, which drive actually the process of transition. And if you inhibit those with maybe a very subtle treatment, not those harsh chemotherapy which you have to do at the end, but very subtle treatment in the beginning, in the early stage, you can actually stop this process and maybe reverse it. So that's a hope. But that's something, a very interesting topic, I believe. Okay, another question, how recommended workflow to analyze methylation data? So again, as an online tool and recommended workflow, again, I would recommend to use our platform. And in the platform, in the platform itself, I showed today the GeneXplain, the Genome Enhancer. Yes. Here, the Genome Enhancer tool, which does everything for you automatically. It's actually a big pipeline which does the things automatically with all kind of embedded parameters. If you want to do it step by step by yourself, it's also possible by doing it in the platform. And the platform is a collection of all those tools, which then at the end are put in the pipeline. And here we have several pipelines already, smaller pipelines, not such a big pipeline like in the Genome Enhancer, but smaller pipelines in the epigenomics part of geneXplain platform. And you can load your data, you can analyze genomic intervals. So if you have already your methylation data already transformed into the intervals on genome, you can load them and analyze them. If not, there are tools here which would which would load your CPG data and then transform them into intervals. Then you can do the functional enrichment like gene ontology analysis. Then you can do the analyze networks, and all of these tools is actually pipelines. You can even see how this pipeline look like. So it has a number of tools which are blue boxes here, and they are connected to each other. First you read your data, then you run analysis of sites, then you compare it, and so forth. You can actually modify those pipelines as well. So that's a very flexible platform to do this kind of analysis. All right. And I have another questions here. Wow. Thank you for your very active contribution. So, okay. So first question, can the geneXplain tool help develop predictive models to assist in forecasting the probability of developing oropharyngeal clefts in individuals based on the set of complete genomes? All right. I don't know what is over oropharyngeal clefts. I will, of course, look at that. But okay, there's, I guess, some kind of rare disease. And what you're talking here is about genome sequences, right? It's not about epigenomics, not about DNA methylation, but genome sequences. And that's a good question. Okay, it's not for this coffee break. I have to check if we had a previously coffee break devoted to the genome sequences to genomics. But indeed, in geneXplain platform and also in Genome Enhancer, you can analyze genomic data. You actually can load here the sequences as a FASTA, as you said, this is just FASTA sequences. And do the same analysis, or you can, if these are genomic data, and from a human genome, and positions of mutations, you can get this VCF file, this VCF, which tells you where the mutations are. So you go here in genomic variants and you analyze them here. And again, here there are tools which can find which binding sites for transcription factors are affected by those mutations. So please try it. It's very interesting. Another question. Right, that's about this G quarters or G force are there are where the sequences on one strand is something like GGGG, okay, or can be GG, blah, blah, GG. They can form G quarter structures if the base preceding this GG is a C on the upper or lower strand, then a C might be methylated and might affect the quarter formation. But one would have to search for literature to see. All right, I see. Well, I wasn't aware with special structures. As I understand, they form a special space structure, although I guess it's only done if the DNA is naked, right? If you have just DNA in the solution, then these kind of structures easily can happen. But remember, in the cell, those DNA is covered by a lot of chromatin, other proteins, which may actually affect those structures. Although when it's opened, when the chromatin is away and you have just naked DNA, they can influence from cockpit. Interaction between G quadruplex and DNA methylation formation and stability. So there's some papers. Thanks a lot. I will look for these G quadruplexes. Probably they have a interesting role. Thanks a lot. So there's all about this quadruplexes. I will check that. I guess it's a very special topic, and maybe not many people are really interested in that among our listeners. So, but I will copy this information which you sent to me, and will check that. Also, although you're anonymous, but if you would like to send me email, and we can discuss this a bit more. All right, thanks. Okay, so in the last question, how to integrate other omics data, RNA-seq proteomics, together with methylation data if available? Bingo, that's important question, and that's something I didn't touch today, although I mentioned a little bit of combining expression data and methylation data. As I said, in Genome Enhancer, we made an effort actually to combine all kind of omics. So I have a nice slide about that, and that would be the last slide, I guess, today. So you see here all kind of omics here on one slide. So we have transcriptomics, we have proteomics, we have DNA methylation, although also a toxic, open chromatin, even interactomics, genomics, mutations. So all of these omics is kind of a different points of view on the same process. So, and the processes is in fact what happens in the cell from receiving a signal and then transducing it to this nucleus, activating transcription factors, which would activate the genes. They will be translated, transcribed and translated. And then those enzymes will do some metabolism in the cell. So all these kind of data are actually different angles to the same process. And that's why just mechanically we can combine them together to reveal those master regulators. And that's how we program this Genome Enhancer tool, where you can drag and drop here transcriptomics, epigenomics, proteomics, genomics, metabolomics, and that will be mixed together in this process of revealing master regulators. But that's a big story, so let's talk about that in the next time, and about the combining many different omics together. All right, so I think we have all the questions for today. And sorry for being quite long today. So we have more than 1.5 hours. And thank you for your attention, and thank you for your questions. And come back to the next coffee breaks. Thanks you very much. And see you later.